and I forgot the brush. So those steps are very similar to how you would analyze your MLST data. So for Melva, it's also importing, have the software determine the copy numbers, and then analyze your data. So, because it can be highly automated, many different isolates can be imported at the same time. So the data set is created. Then for each VNTR, there is a fingerprint type created. And then all the lanes are imported and assigned according to that fingerprint. So here you only have a fingerprint type per pool and die. And then you need to assign the copy number, so you're working with raw data, which is the next step. And then for that, you need to provide some parameters. So the pool and die, if you're using such a strategy it belongs to, you need to enter an offset. So where does that VNTR start? The repeat length. A minimum and maximum expected copy number. That's just to speed up the analysis so it doesn't keep searching forever. And then some position tolerance. So you need to do that for all the VNTRs. So for example, if you do the Miro VNTR, you can either work with 15 or 24 loci. So it is time consuming, but you only have to do that once for that experiment. So for each VNTR, then the software will look at all the feasible bands. And how does it do that? It will look, so it has to fulfill this formula, so that your offset plus the copy number times your repeat size minus your observed band size is less than your tolerance. So software will do that for you, and what you will get afterwards is a calculation report for each sample and for each VNTR. And what will it give you? It will give you the copy number. It will also give you the observed size. Below that, the theoretical expected size and your peak height. That peak height is important, that if you have multiple peaks, so stutter peaks or that bleed-through peaks, that you see that the highest peak is chosen. So how do you analyze that data? The coefficient you would choose in most cases is a categorical coefficient for copy number data. And the reason behind that is because one single mutation may introduce or delete several copies at the same time. So 4 is not closer to 5 repeats than 5 to 10, for example. It can be that it's just one single evolutionary event. So what is the most common way of analyzing MLVA and MLST data? Is by use of minimum spanning trees. Minimum spanning trees are also the trees that are used, for example, in the burst algorithm and eBurst if you do that online. They are known for a long time in mathematics and other areas. And it's a very simple assumption that these trees make is given a set of distances or similarities between your samples, the minimum spanning tree will be the tree that connects all your samples in a way that minimizes the distance of all the sum distance of all the branches. So it looks at the simplest explanation of your evolution. So minimum spanning trees are often confused with parsimony trees. Now they share the same idea that your evolution should be explained with as little events as possible. So the most likely evolution is the ones that is the simplest, which is basically Occam's razor. However, if you use parsimony, you have some hypothetical nodes, whereas a minimum spanning tree in its purest form just looks at all the states that are in your data set. So if you did not sample enough, it will not introduce a hypothetical node here if it decreases the distance, for example. So to give you some pros and cons on minimum spanning trees. Minimum spanning trees should not be used for phylogenetic purposes because they should be used only in studies that focus on a very short evolutionary time frame. Why is that? Because those hypothetical nodes will not be created. So it assumes that all the states are present in your study. So your population that you take should be big enough. So the sample data should be complete enough. So it represents the full biodiversity of the forms or states as closely as possible. Minimum spanning trees can contain some star-like branches, and that allows for correct classification of population systems that have high mutational or recombinational rates. So minimum spanning trees are really good for microevolution, not for macroevolution. Population genetics, just looking at a certain population at a short period of time, and also at epidemiology. And it's very adequate for analyzing MLST and VNTR data because you can use a categorical coefficient on these techniques. I didn't, in your handouts there are more slides, but I linked a little bit. There are a bunch of slides that explain the priority rules that are used in case that there are possible same states. I'm not going to go over that. But you can read that yourself. So as I mentioned earlier, when talking about UPGMA, there are other algorithms that can be used as well. For example, complete and single linkage. What is the difference between UPGMA, single linkage, and complete linkage? Is UPGMA will average out the remaining part. Single linkage will take the highest value. Complete linkage will take the lowest value. So they can have some very interesting properties in epidemiology or population studies, especially if you are not using similarity values, but different bands, for example. So if you use complete linkage together with a number of different bands, then you can cluster all the patterns that have maximally, for example, 2 bands difference with any other. Or if you use single linkage, where you look at the highest value, then you cluster all the patterns that are identical with at least one other pattern in that group. So there is some relevance on that different number of bands. The first case, if you're using electrophoresis fingerprints, so you're looking at distance or different number of bands, and complete linkage clustering. What is the result? Those are groups that are guaranteed to be no more than, for example, n bands difference apart. So these 2 groups, so this whole cluster, they are just 1 band, maximum 1 band apart. So this, in outbreak detection, for example, it can be very useful. which is something you cannot see with UPGMA. For MLST, if you're looking at the alleles, so not at the concatenated sequence, but at the alleles, and you use single linkage, then the result is your exact representation of what the clonal complex should look like. So those were two other ways of looking at your data, which in an epi context can be more meaningful than, for example, always sticking to UPGMA and your DICE. Then, because you are, or you can do different techniques, you can also compare the results from those different techniques with each other. There are two ways that I'm going to discuss here in short, which is congruence of experiments. What is a congruence of experiments? Basically, is you will compare the pairwise results of your different techniques. So, what is your question there is, do they tell me the same about my data on the same taxonomic level? Yes or no? If they tell you the same, then you can already think, do I need to do those two techniques? Because if they tell you exactly the same, why bother doing twice the work? And then you can make another decision. If I have one technique which is time consuming and very expensive, and you have a faster one that is less expensive, okay, stick to that last one. So that's another way of using that congruence of experiments. Another way of comparing results from different experiments can be partition mapping, where you are, for example, comparing different classifications with each other. You can compare, for example, Miru and Spolio, but what you can also do is compare a very old technique, phage typing, for example, with MLST. And see if you can, for example, predict, based on your MLST pattern, your phage types, which basically you can do. So, the first way of looking at different experiments is the congruence of experiments. And then for your multiple experiments, a correlation similarity matrix is calculated, which is shown here. And this is just turned into some kind of dendrogram of experiments. And then as an added value, you can add some standard deviations and significance on the correlations between the techniques. What can you learn from this? Firstly, you can see which techniques are very similar, which techniques are very different. But for example, if you would look at 16S and DNA-DNA hybridization, you could learn something more. And that is, for example, that you have techniques that very badly distinguish closely related sequences, according to, for example, 16S. But another technique could have a better discriminatory power on that taxonomic level. And that's something that you can see from that curve. So if you have isolates, all these gray dots are in fact pairs of entries. So it's pairwise comparison. So all these ones are more or less the same with experiment 1. But they can be distinguished between, I don't know, 30 and 100% with the other technique. So that gives you some proof of your discriminatory power and on what taxonomic level of your technique.